What is that one thing you guys remember in that pathway that is very different than any other pathway? It's different.
In all the other pathways, that itself is modified, but in this, the inhibitor is restricted.
Okay, that is one striking difference. Yeah.
Also, the degradation is causing activation of the pathway.
Right. These 2 points are complementing. But something which I remember is K63 cells acting as a scaffold to recruit the effector and the kinase together, right? TAK is the kinase which will phosphorylate your, which one? IKK beta. IKK beta, so that it can go and phosphorylate your inhibitor, as Zoya said. Good. Anything else we discussed? Sign? Of what? There are 2 types of chains, right? K48 and K63. Good. And Nemo is the guy who can go and recognize the K63 chains. Perfect. Anything else? I think it is, that's what it was, no? Perfect. Okay, so today we will talk about notch pathway. Do you guys know about notch pathway? Anyone? Have you heard about this? This is a perfect example of that paracrine signaling, maybe where 2 cells next to each other will talk to each other. So what do we know about notch pathway? Have you heard? You were saying something? You forgot. Okay, I thought he said. Anybody else? Do you know? Sahili? And the receptor. How are you, Durgesh? I asked how are you. You are not ligand. You are good? Perfect. Now tell. Very good. And notch is the receptor. Perfect. Good. So essentially this pathway is very important for the stem cells, essentially self-renewal and de-differentiation of stem and progenitor cells. The classic examples are neuronal stem cells or multipotent progenitor cells. So this notch pathway is very important in these signaling, in these kind of cells. Also it is involved in selection of cell fate, and it induces fixed differentiation of progenitor cells, such as differentiation of cardiac progenitor cells or hepatoblasts into bile duct and lineage cells. So whenever there is any injury, let's say in there is liver, there is some injury and all, right. So it will stimulate the proliferation of some of the cells, which is going to help in the renewal or regeneration or repair of those tissue. That's why your notch pathway is very important. And in fact, you can take, there are so many diseases also where it is dysregulated or deregulated. One example they have given, like for example, you look at it in these hepatocytes, there are this BEC, right. So you can say that bile duct epithelial cells will be there. So if there is any injury, what you need to do is you need to really amplify these cells. How do we do that? So essentially the notch pathway gets hyperactivated or activated in your hepatocytes. Hepatocytes will be, those are present in the liver cells, right. So they are present over there, and then it induces the differentiation of hepatocytes into these biphenotypic cells. Biphenotypic maybe because they show the phenotype of both hepatocytes as well as your bile duct epithelial cells, and eventually they will differentiate into formation of bile duct epithelial cells. So this is one example of how notch pathway functions in a particular context. So in Drosophila, I think notch pathway was discovered several years ago, if I correct, 100 years ago or something. There was a first paper published in 1916 about the notch pathway, and then it was studied in C. elegans, which is this nematode worm, right. So it has been studied there as well. So in Drosophila, when it was studied, they identified that there is one notch receptor and there are 2 ligands to it in Drosophila, the transmembrane proteins. And interestingly, both the receptor and ligand both are transmembrane molecules in this context, whereas in most of the other pathways we saw your receptor is only the transmembrane, and then ligands will be, maybe they are lipidated or whatever, they are secreted out, right. In case of hedgehog or Vint, we saw that they get modified, but they are secreted outside. And then there are 2 ligands which are transmembrane proteins called delta and serrate. That is in Drosophila, whereas in mammals we have 4 notch paralogs, notch 1, 2, 3, 4, and there are delta like DLL, DLL1, 3 and 4 and jagged. You must have heard about these names sometime, right? Anyone? Aniket, have you heard these names? Right? Have you heard about it, Pratibha, any time? No? First time? It's okay. It's first time, it's no problem. It's not that we all of us we study, right? Have you heard about this? Not jagged, but notch, delta, this one we know. These are very commonly, maybe because they are studied in Drosophila also, that's why we know. Something very interesting and very important to make a note of is that ligand mediated activation of this pathway, it leads to the series of proteolytic cleavages in the membranes of notch family of receptor, which release the notch intracellular domain. So, we will see how exactly the pathway gets activated and what will happen, but this is slightly very different than your conventional pathways, whatever we saw, whether it is RTKs, JAK-STAT, or for that matter we saw hedgehog or Wnt or GPCRs and what not. There we had a ligand that comes, binds to the receptor, receptor gets activated, maybe transphosphorylation happens, then there is a downstream phosphorylation of some other molecule, happens, right. Then in case of Wnt and hedgehog, we saw that the key molecules, they are degraded when pathway is inactive. The opposite of that was NF-kappa B, when upon the pathway activation something got degraded. So, these things happen. In case of notch, there is role of proteases that we will see a specific family of proteins, they do play a role into this, and the cleavage events happen. That is how the pathway gets activated, which is going to make something, a product of these cleavage events is going to be the molecule which has some transcription activity, which is going to go and do the job for it. In rest of the cases, we had a defined transcription factor, whether STATs or NF-kappa B pathway we saw, right. There was some SMADs, or in case of RTKs, there are multiple transcription factors which could get activated and what not. In this case, it's slightly different. So, this notch intracellular domain, NICD, you guys can make a note of this. This is the guy, this is the domain which will have this will have the transcriptional activity as well. So, what happens? You have the whole notch ligand, then there are proteolytic cleavages happen, notch receptor, the ligand binds to it, it gets cleaved, and then the C terminal or the cytosolic, this NICD fragment, it gets released from the path, the whole molecule, and once it released, it directly enters the nucleus. Okay. Once it enters the nucleus, what it does, it goes and binds to these molecules, which is CSL, that is CBF1 suppressor of hairless. So, most of these names, they have been given based on the phenotype what they observed in Drosophila. You guys remember the what is that called as wingless or hedgehog and all these things, based on the discovery they have given. So, that is very typical of them, because this pathway was discovered in Drosophila, whatever they find and they give the name, like suppressor of hairless, and this is a DNA binding protein, plus there is a co-activator called mastermind. एकदम खतरनाक वाला नाम दिया है उन्होंने, mastermind, which is mastermind like transcriptional co-activator. It's a transcriptional co-activator, not a transcription factor itself. So, these two guys come together and they will stimulate the transcription of target genes in case of activation, in case of this notch signaling pathway. Remember, NICD is very important. So, something to make a note of, and that is very important, is that this NICD, once it gets released from the main receptor, it directly enters the nucleus. There are no intermediates. If you go back and try to remember what happens with your SMADs, there are I SMADs, R SMADs, O SMADs, where they have a SH domain, right. So, there is this kind of structure they form, the receptor gets phosphorylated, this R1, R2, R3, they come bind to the ligand, get phosphorylated, it will phosphorylate the SMAD. Then O SMADs are the ones, or they get 2 and 4, they come together, uska nuclear localization signal will get exposed, then they go to the nucleus and they do it. So, there are too many events involved it, right. Similarly, if you take any other molecule like your stats for example, right, the cytokine receptors, they do not have any catalytic activity. You remember, right, these are the exceptions to every pathway, like your RTKs, HER2 does not have catalytic activity, but it has the ability to bind to the ligand and homodimerize or heterodimerize, whereas HER3 has the activity and then they can come together, heterodimerization. You remember we talked about these things, remember Rutuja, right? Good. So, in this case there is no intermediate like that. You have NICD, which is the transcription factor, it has the ability to act as a transcriptional coactivator, it goes directly to the nucleus. So, because of that there is no amplification of signal happening. In case of G protein coupled receptors, you remember we did say, right, how does the amplification happens? It happens through your cyclic AMP or calcium signaling and what not. In case of RTKs and all, you can imagine the amount of receptor will get phosphorylated, those many molecules. Some of those kinases, they have 150 different substrates, right, they can go and phosphorylate those molecules. It depends, there is a signal amplification would happen, but here there is one-on-one interaction happening, one ligand, one receptor coming and binding together, only this fragment is going. So, that is why there is no signal amplification happens, which is different than any other pathway, and that is when the NICT takes responsibility for implementing the pathway activation. Is it clear? Remember, these are the striking differences between this pathway versus any other pathway. So, if you look at it, this has structurally this is how they look like. So, these are your ligands which are the sending cell. So, the cell which is having signaling ability, they will express the ligand, whereas receptor will be present on the responding cell. So, when these molecules were discovered in 1916 and when they were cloned, there were some papers, I did not pull out the papers, but when they were cloned and discovered, they identified that these molecules, whether it is delta or the serrate, they have something called as EGF repeat. So, it is epidermal growth factors, right. So, some of these transmembrane proteins or the receptors, they do have these kind of domain architectures where you find the EGF repeats consistently, and they have 1 to 17 repeats. In fact, the receptor also. So, that is how they have very long extracellular domains, and these repeats will be folded in certain conformation. They do have something called as cysteine rich domain, which is missing in delta, but serrate has it. That is just before your EGF repeats, and then they have DSL, which is delta serrate LS LAG2, and NT is N terminal or amino terminal domain. So, this DSL and NT, these are very important to go and interact with the notch receptor. So, if you look at it here, this EGF repeat 11 to 12 of your notch receptor, you see orange color fragment whichever they have given. So, these guys DSL and NT, they go and associate with this receptor at this 11 and 12 EGF repeats on the receptor. So, this is the domain architecture for your ligands, okay, which is your serrate and delta. Similarly, your notch receptor itself has some domain architecture. So, the important thing to remember is this notch receptor, it is there are two heterodimeric portions, HDN and HDC. So, this one HDC and HDN, these are the two regions which are bound to each other, they are very important, they interact with each other, and then they have this something called a cysteine rich lean 12 notch repeats, which LNRs, which is just present next to this HDN. So, these three guys, when they are together, HDC, HDN and LNR, they will form this negative regulatory region. Remember this is very important, NRR. What is negative regulatory region? We will see why that is important. This is made up of your HDN, HDC and LNR. I would suggest if you guys can remember the full forms of all these, it is good, but to the least understand the function of each one of them, that is very important. Okay. These three guys come together to form a structure which is called as NRR, that is negative regulatory region, which occludes. So, I will show you next slide what is the importance of this NRR region, but remember that. Then they have something called as notch intracellular domain, that is NICD, which was this case. This thing which was very important, which is this one, which is present in your inside the cell, and it has this domain called RAM domain, which is RBPJ associated module. I am really sorry, I do not know what this RBPJ means, but remember that it has this RAM domain and anchoring repeats, these A and K, these repeats. These are again some of these repeats, some of these proteins, these are structural domains that you will find in so many different types of proteins. They might have scaffolding function or protein-protein interaction functions. That is how these repeats have been there. So, this molecule, your notch receptor, again it has very long EGF repeats at one side, which is present, and then out of this whole EGF there are around 36 EGF repeats. Out of that 2 are very important, 11 and 12, because they associate with your ligand, and in ligand there are 2 molecules which associate with these 2 repeats, right. And then there is NRR, which is made up of HDN, HDC and LNRs. So, NRR is very important, why and how, that also I will tell you in next slide. And then we have RAM, which is the intracellular domain, that is inside the domain, inside the cell. Okay. These along there is anchoring repeats, which is required for interaction with the DNA. You remember I said no, this guy will go initially itself, we said no, this NICD will go and interact with this CSL, which is a DNA binding domain. So, these repeats or these structures are very important for its association with the DNA binding domain, because that is how it is going to go and act as a transcriptional activator or co-activator. Point A. Point B is that these anchoring repeats, some of them, they do have nuclear localization signal, because you remember in this whole pathway there is no intermediate. The moment your this NICD gets released, it will enter the nucleus, which means that it should have a nuclear localization signal, right. It is not, which will be mostly through importin mediated nuclear localization. So, that is also present over there. So, this is the molecules which are present in that. Now, coming back to this your NRR, why does it so important? So, when and then there is PEST domain, which regulates the NICD degradation. These PEST sequences, sometimes they are important. I think it must be going through ubiquity independent degradation. I do not recollect any specific reference for that, but it does regulate its stability. So, this is how in general it goes. This is your signaling cell and this is your responding cell, right. So, this is your delta, which is a ligand, and this is your receptor. This is your NRR, and then we will come to the point where which I mentioned that there are cleavage events which happen. But if you pay little bit close attention, the binding of this delta, which is a ligand to the receptor, is going to create something into this. So, when the pathway is inactive or when the ligand and receptor are not binding to each other, somehow this NRR, it is called as negative regulatory region, because it somehow hides the protease sites, the sites at which this ADAM protease is going to go and cleave. So, that this protease will not cleave the receptor, the notch receptor, when the receptor is inactive, when it is not bound to the ligand. Are you guys understanding what I am saying, right, Durgesh? So, what happens the moment your receptor, so this is how the arrangement is. You have this EGF space, you see this NRR, you have LNRs and HD and LDN or HDN, what are the names here, no? HDN, HDC and LNRs. You look at this LNR, you look how the conformation is, right. It is kind of shielding it like this, so that your protease cannot go and access. So, this is a protease ADAM which will go and cleave somewhere over here. So, the cleavage site is shielded by this region, that is why it is negative regulatory region is very important. Now, what happens the moment your delta comes and binds to this, there is some kind of pull will happen, it will pull it. The moment it pulls it, this NRR will open. The moment it opens, then your site gets accessible to protease and then protease can cleave it. So, this mechanism is very important to remember. That is how you can keep a check on your activation or inactivation of the pathway. You guys following what I am saying, right? Imagine if there is some mutation in this NRR molecule, what would happen? It will not be able to shield the protease site. Then if it cannot shield the protease site, maybe your atom can, this atom 10, there is a metalloprotease that can go and cleave it. If it cleaves it, then you will have a fragment which we do not know what it would do. So, this is part of the regulatory mechanism that only when you need it, you activate it, and your ligand binding makes it very important because it is going to attach and then it will pull it, so that it opens up. Is it clear? So, this is part 1 of this pathway. Actually, yeah, this is part 1 of our pathway. The second important thing is matrix metalloproteinases. Have you guys heard about this name any time? How many of you? You have heard? What do we know about them? Mechanical cues, okay. Anybody else? What else? Cell migration. Central metastasis. Invasion, yeah. That's it. It's good thing that you guys already know. Then matrix metalloproteases, the name itself, the metalloprotease is nothing but the metalloprotease. It's a protease which is dependent on some protein ion. So, here they need zinc. So, these are zinc dependent endopeptidases. It's very important. What do we understand by endopeptidases? It can go and cleave anywhere. They are not exo, right? Side से करेगा, endo बीच में जाके काटेगा, कहीं भी काट सकता है. And they are zinc dependent. That is very important. And they play critical role in degradation and remodeling of extracellular matrix. If you guys get a chance, you can see a work of Professor Shamik Sen in our department. He works on these metallo MMPs, right? MMP9, MMP12 and all that. I don't know if he teaches this course also about mechanical. Yeah, you guys are taking that? Okay, so you already know what it is. So, the family of zinc dependent endopeptides. So, they are metal dependent, but they do go and remodel the extracellular matrix. So, there are 3 subfamilies. One is matrix metalloproteases, which is MMPs, a disintegrin and metalloproteases, ADAMs, and ADAMs with thrombospondin motif. I am not from this biology background, but essentially what is that there are different families. All of them, they come a bigger umbrella of metalloproteases because they are dependent on some zinc ion or some metal ion, but based on what they do, like there are certain differences in their function. Now, this is the structure of ADAMs. Now, we are going to talk only about this ADAM family of metalloproteases, which is this second one, a disintegrin and metalloproteases. So, this is the general structure. Oh, I forgot to put a reference here, I am sorry about that. So, I'll put it some nature review. So, this is the general structure of these ADAM proteins, where you can see that it has some convertase recognition sites, and then this is your metalloproteinase domain, which is zinc dependent. That is how it is going to go and cleave the molecules. It has this disintegrin domain. I understand maybe it has something to do with integrin association. I think some of these metalloproteases do go and bind to the integrins, which are very important to maintain the cell-cell junctions or the contact between 2 cells. Then it has this cysteine rich variable region. Then there are EGF like repeats, or which is also called as stalk region, and this is the transmembrane domain, and these are single pass transmembrane proteins which are always associated with the membranes. And these are the different ADAMs, ADAM 8, 9 and what not. So, you can see this is their domain architecture. Of course, they share the similar structure. The only thing is, in case of ADAM 10, which is the molecule of our interest for this particular context in notch signaling, it does share most of the domain architecture like zinc dependent metalloprotease domain, then this pro domain, and then it has this disintegrin domain, but it does not have this EGF like repeats, it lacks them, which is again this region between the transmembrane domain and the variable region is called as the stalk region. That is what they have. So, this is the molecule of our interest, ADAM 10. So, this belongs to this metalloprotease family. So, what we saw so far in notch signaling pathway, we saw the ligands. What is the ligand? In Drosophila it is delta or serrate, and in mammals there is jagged and delta. So, we saw the ligands, how is the structure of the whole receptor or ligand is there, how does the binding of ligand will expose the proteolytic site, so that this ADAM10 protease can access it. This is the general information about the pathway. Now, let us see the pathway as such, how does it work. This is notch delta signaling pathway. Actually, this is the only thing I thought I will teach you guys today. It is very short, should have gotten some more. Can everybody see this? Yes, everyone can read it. Durgesh, can you read? You cannot read anything? Oh, this is not that clear, but you can see the structures at least, right? At the end, everyone. Saheli, can you see that? Good. I will take you guys through this. So, now as we said, there is a responding cell which is this lower panel, and there is signaling cell which is upper panel. Okay? So, these signaling cells, they express your ligand which is delta. This is written over delta over here, and this is your ligand. You guys already know what is the domain architecture of ligand that we saw, what it has the cysteine rich domain and then the interacting region with your notch, and that is it, there is nothing much, and EGF like repeats. And this is your notch receptor. Again, it is a heterodimer, right? That is what we saw, and it has so many EGF like repeats. Then there is NRR, and inside there is this NICD molecule actually, which has anchoring repeats, PES sequence, and there is one more thing. RAM. So rare hoga. Anyways, it's very straightforward though. So we have this notch receptor over here, and this is where your ligand is there. And as I was telling you, this NRR region is gonna, this LDR, whatever those HDN, HDC and whatever LD, LNRs, right, they will fold and protect the site. That is what it is happening. This is how the orientation is. ADAM10 is single pass membrane, it is already present in the membrane, right. The moment your ligand and the notch receptor and ligand will come and bind to each other, it will create that pull so that it gets exposed. That is the only thing happening. And then your ADAM10, which is green color enzyme over here, it is going to go and cleave this receptor. Once it receptor cleaves, this guy, the ligand bound to this extracellular region of notch will be endocytosed by the signaling cell, and then it will get degraded. I think it will get endocytosed and it will follow the degradative endolysosomal pathway. That we need not to bother. We have to think about the responding cell. Now you have this teeny tiny space, this membrane associated fragment of your notch receptor, right. It is not, it still has this NICD sequence, but there is another complex which comes, which has gamma secretase, preselenine 1 and nicastrid. So, there are 3 proteins which come. Again, gamma secretase is one of those, I think it has the peptidase activity. So, these 3 molecules will come together. So, there are 2 cleavage events happening over here. First is by your ADAM10 and second is by your gamma secretase complex. So, first ADAM10 comes, cleaves it, the ligand bound extracellular EGF repeats, this portion will get endocytosed into the signaling cell, that goes away. Then the fragment which is membrane bound, which has your NICD along with it, it will become substrate for your secondary complex. This is the complex which has gamma secretase, preselenine 1 and your nicastrin. So, these molecules, again it will make one more cut, and only the intracellular region, which is your, now what is remaining is that NICD. NICD will enter the nucleus, it goes and associates with this DNA binding domain which was CSL, and then there was mastermind which is the co-activator which associates with that, and it removes these co-repressors. The COR is the co-repressor, it goes and sits at the DNA binding elements along with these guys and drives the expression, whereas the co-repressor will be kicked out of the DNA, and that is how the pathway works. It is very straightforward pathway, there is nothing complicated about it. All these pathways are very straightforward, there is nothing much to remember. Few names, but I think it is ECPC. What happened, Durgesh? More names, do you think? Huh? But it's very easy. I mean, you guys have never heard these domain architectures before. The EGF repeats, maybe that's what makes it difficult. Nobody worked with notch signaling, no? I don't think anybody works also. It's difficult to study. We don't have Drosophila lab in our department, neither in chemistry or chemical. No, nobody was Drosophila, right? It's very interesting model though. You guys work with yeast. Did you guys understand this? So there are three things which are very important here. First thing is there is a role of metalloproteases. The whole pathway is regulated by these metalloproteases, ADAM10 and gamma secretase. These two are very important. That is first thing. Second important thing is NICD, that is the fragment, the output of two cleavage events is your NICD, which goes and acts as a transcriptional transactivator along with the DNA binding protein. And the third most important thing, Durgesh, what is the third most important thing to remember about notch signaling? हाँ, वो पहले बोला मैंने, वो पहला था. What do you think? What is the third important thing to remember? NRR नहीं, extracellular नहीं. There are no intermediates. There is no signal amplification happening. It is one on one. एक ligand, एक receptor, एक activation. That's why it functions in the niche areas. The stem cell को activate करना है, then it will go and activate only because it acts as a paracrine signaling, right? These are juxtacrine, whatever you call it, that those two next to each other, they will talk to each other and get done, the job will be done. And this is very important in context where we understand the role of metalloproteases and how it is going to create some of the other molecules which are signaling relevant, right? Your NICD is the product of two cleavage events, right? Imagine something goes wrong with them, what will happen? हाँ, Sudha, we don't know what all can go wrong. There is one possibility that NICD is never made. If your catalytic activity of ADAM10 is gone bad, then it will affect your hepatic regeneration. No? Can you think something like this, Shine? Oh, part 2. You understand what I am trying to say, right? So this kind of questions I will ask you guys in exam. ऋतुजा is like गए यार, क्या यार, ऐसे वो model question paper was good. No, you have to think. Say for example, you can identify, somebody might have identified a mutation in your, what is that called as, whatever, leucine repeats, whatever we see this, no, LDR sequence here. This one, LNRs. if there is a mutation in that LNR, what would happen? No, if there is a mutation in this LNRs, what would happen? Ligand? Ligand? It will not be able to form this NRR, no? It might not be able to, it will not, we are not sure, I did not do the experiment. There is a possibility that it will never be shielded. Then your ADAM10 can constitutively cleave it, whether your delta is there or no. We do not know. If the mutation is going to affect the, it's going to, there are two possibilities, right? The mutation will either make it accessible, or it might bind so tightly that even if it pulls, it will never open up. Then the pathway will never get activated. You understand?
NRR is a negative regulator, so if some of its domain is getting mutated, so that means the not signaling will continuously get activated.
That is true, but mutation बता के नहीं आएगा ना कि वो ऐसा ही करेगा. See, any mutation can have two fates, or three mutation can have three possibilities. I am talking hypothetically. Either it will not change anything, it will be like your silent mutation, or gain of function or loss of function. तो उस हिसाब से कुछ भी हो सकता है. I am talking hypothetically, I do not know. It is NRR, they named it because it does that job, but mutation कुछ भी हो सकता है. So these are the three possibilities. So you guys will have to think about these kind of thing and then write an answer to the question. Anyways, now let's think about there is another pathway which all of you guys know. Have you guys heard about this Alzheimer's disease? Everybody knows? Who knows, who doesn't? Nobody knows about Alzheimer's? Huh. What is it? Neurodegenerative disease. Thank you. Plaque. Misfolding of the protein. Anything else? Amyloid formation. Gaurav? बीटा स्ट्रैंड स्ट्रक्चर किसका? अमाइलॉइड.
It's hard to study.
Hard to study. Do we know why, like what is wrong in these pathways which could lead to the formation of these oligomers or plaques? This pathway is already in front of you.
Wrong cleavage.
APP you guys have heard, right? APP, amyloid precursor protein. I thought I did not to tell you, guys will be telling you, because neurodegenerative disease is sort of fancy है ना, everybody, people, cancer, neurodegenerative diseases. So APP you guys don't know, amyloid precursor protein. So you don't know the full name. APP everybody knows, amyloid precursor protein. So this one also, there is the role of these metalloproteases. So usually what happens, this APP which is blue color, this protein over here is a transmembrane protein. This ADAM10 or ADAM17 which is alpha secretase, again these are the molecules which will get secreted and then they are gonna go and cleave some substrate, right? Your ADAM10 was going and cleaving something in this notch receptor, so that the next molecule gamma secretase complex comes and does it. Here what happens, they have different cleavage points, right? So this alpha secretase ADAM10 or ADAM17 will go and cleave this APP over here, then this short fragment of APP becomes the substrate for gamma secretase, Again same molecules, penicillin, gamma secretase and all these guys, they come, they make one more cut. Once they make one more cut, you see there is this peptide membrane associated peptide of 26 amino acids, right. One is 14 amino acids, one is 12 amino acids. So, this short fragment of 26 amino acids, it is not, it is not harmful to the cells that is formed. Okay. Cells do not bother, I mean, ho gaya hai, theek hai, no problem, everything is going fine. The moment your APP gets cleaved by this beta secretase, which is going to go and make a cut at the different place than your alpha secretase, and then this becomes substrate for your this gamma secretase complex. So, essentially it is going to go and make the fragment will be of 42 amino acids, which is 28 plus 14 is 42 amino acids, right. And these 42 amino acid 42 long amino acid long fragment is your A beta, and that is what they have inherent nature to undergo oligomerization, and then that will go and form the plaques. That is how your something goes wrong, it gets deposited, the what is called as neuronal toxicity will happen, the cells will die and what not, and everything will happen, ultimately giving rise to this disease condition. So, this is another pathway where your potolytic cleavage becomes very important. So, both pathways, whether it is a notch or this signaling pathway, whatever, it is not a signaling as such, but this is a pathway which is relevant to health and disease, where your metalloproteases like ADAM10, which essentially you have to remember alpha secretase and gamma secretase complexes, beta secretase is not there, alpha beta secretase, both the complexes are, these complexes become very relevant, alpha beta gamma secretase complexes, where there are metalloproteases, they are processing some of these molecules. As such, I think APP has some neuroprotective role, that is what people know, but even if it is not doing anything, it is not harmful to the cells. But when you cut it at wrong place, it is going to create some peptides which will go and create trouble for, it will get oligomerized and then forms the plaques, and essentially it will lead to the neurodegeneration. So, these both pathways have something common in them, that is a function of metalloproteases. So, this ADAM10 family or these MMPs are very important in case of context of cancer and extracellular matrix remodeling. So, that as most of you said, the cells can, cancer cells can undergo EMT, that is epithelial to mesenchymal transition and migration, that is how they are important. But this ADAM family, which is a disintegrin and metalloprotease family molecules, play important role, whether it is not signaling where they important in the cleavage of the notch receptor, or in this case APP processing as well. If APP after ADAM-TS, ADAM10, if the peptide becomes a substrate for gamma secretase or beta secretase, that is how the decision, instead of ADAM, if it becomes gamma secretase, gaya kam se, then maybe the gamma secretase will have some important role to play, and that is how the disease might happen. Is it clear? This ends the notch and little bit of your APP processing pathways. So, we are done with most of the signaling pathways. The only thing remains is RAS MAP kinase pathway, which we will finish in next two classes.